is. You have some perspective on that. So obviously I won't get through all this today. What I want to talk about is several different types of tests, and let's look at the themes. So the themes are excessive divergence over time, excessive divergence over space, and then looking at a pattern along a chromosome to see if that pattern of variation is consistent with the sweep. So I'll give an overview of polymorphism-based tests. I'll then talk about allele frequency tests, Whipple's adjusted, the Fisher-Ford test, the Schafer linear trend. I'll talk about McDonald-Kreitman tests. They're what I call McDonald-Kreitman second and third generation tests. No one uses McDonald-Kreitman anymore, but third generation tests I think are fairly powerful. We'll talk about allele environment associations. That's a modification of the McDonald-Kreitman type approach, and it simply asks, are certain alleles overrepresented in certain environments or not? So polymorphism-based tests depends upon strategy. I could sample a population at 2 or more time points. I could sample a series of populations at a single time point. I could do both. Those models haven't been developed, but they'd be pretty straightforward. Or I could take a single sample from a population. So obviously, least work, more work here. And the other feature about this is if my sample isn't from a sufficiently long amount of time, I may have very little power. If the selection is small, I'm not going to see a lot of change over a period of time. So just kind of going through, so excessive allele frequency change. The first formal test was Fisher and Ford, 1947. But here's a cool idea, is you can use ancient DNA, and here's some references for applying this, and look at allele frequencies in a small sample from individuals which are 3,000 years old, and look at contemporaneous allele frequencies and ask if any of those are excessive. So the cool thing about ancient DNA is you can actually start thinking about looking for signatures of relatively recent selection in humans directly by looking at allele frequency shifts which are in excess. The big problem, of course, is how represented is that ancestral sample? Very often it could be a whole bunch of relatives, in which case your sample is a bit tricky. You have to deal with it a bit more carefully. I could look at excessive allele frequency divergence. There are basically 3 early types of selection that were tested. Fisher-Ford, you have allele frequency estimates asking if they were excessive, 1947. Then you basically had Lewontin-Krakauer, which was I basically compute FST over a set of markers and then look for whether a given marker has an excessive FST. They used a chi-square statistic, as we'll see. That's not really appropriate. Then we basically had the Ewan's Waterson test, about the same time as Lewontin-Krakauer, which is actually using what was endpoint was very crude molecular data. So again, you use the allele frequency divergence is you use FST, and what you can do it several different ways. You can compute an overall FST, then ask if an observed FST is excessive relative to that. You can use outliers. You can use that overall FST to construct a chi-square-like distribution. There are problems with those. You can also then use marker data in third generation tests to generate what that FST should be and use that to more appropriately correct for that. So those are basically multiple populations temporal, multiple populations spatial sample. Then you can have a single population sample, and there's lots of things you can do here, which we'll talk about in the next lecture, like site frequency, allele frequency, LD. For now, we'll simply talk about, we'll talk about spatial patterns of variation. On a chromosome, I have a model that tells me, if I know the recombination rate, how that pattern of variation should vary about my selected site. I can go ahead and ask, does the observed pattern fit not just the depression variation, but the specific shape of that depression and variation? That's a likelihood ratio test. The other things I can do is I can look at that sample from an infinite sites or infinite alleles framework. So I can basically look at changes in the site frequency spectrum. And typically, as we mentioned, those involve computing theta using different measurements of that site frequency spectrum and ask if they're consistent, like Tajima's test. Again, the problem is those are severely compromised by demography or population structure. The other thing I can use is I can use haplotype information. When I say haplotypes, I now move from infinite sites to infinite alleles framework. So I could use, the Waterson test and look at the allele frequency spectrum and ask, for example, is there an excess number of alleles, give my estimate of theta, or if I condition on the number of alleles, is the pattern of variation conditioned on alleles different from neutrality? Of course, the key is different from equilibrium neutrality. Again, equilibrium can compromise that prediction. Other ways to use haplotypes, I could look at distinct signatures in LD that differ from a hard versus a soft sweep. We'll talk about tests for those. Or I can use this idea, and this is a very popular idea now, that a common allele is an old allele and therefore should have shorter blocks of LD. So a lot of the most powerful tests now are based upon looking for long haplotypes, long blocks of LD, and these are often called long-range haplotype tests. These can detect tests that are missed by site frequency and hard sweep tests. So we'll talk about allele frequency, site frequency and allele frequency tests in the next lecture, lecture number 9. For now, I want to talk about basically multiple population samples temporally, multiple population samples spatially, or a single sample using the chromosome pattern of variation. And again, I'll only get partially through. We'll finish this lecture up tomorrow. So one of the key ideas is, how do you attempt to account for departures from the equilibrium model? Not neutrality, equilibrium. The population hasn't been its current size long enough for the rate of removal by drift to balance the rate of input by mutation. The population has expanded. I've got population structure. How do I account for that? Well, Luca Kavi-Schwarze at Stanford noted that demography and population structure should leave a common signal over all the genes in a genome. So if I've got 500 sites, I could compute 500 Tajima's D tests, and those should all be realizations, if they're neutral, about the expectation I should expect for that population demography or that population structure. People then said, oh, you could do that and look for outliers. The problem is, if I generate random curves, I'm going to get outliers. The way to tell if those outliers are significant is to estimate the variance. When you depart from neutrality, you not only change the mean, you greatly inflate the variance. So outliers are very, it's a way, the outlier method basically is a way for people to cheaply find interesting sites, but it's just that, it's an interesting site. It's not a formal statistical test. The second way is to use data from presumably neutral markers to infer the population history. Tajima's D basically is changed if you have population structure or population history. You can then use methods. A good example is approximate Bayesian calculations. You basically assume a certain population structure and simulate that structure. So you draw values. For example, draw the bottleneck size, bottleneck duration from some prior distribution, run the simulations, and then see how summary statistics like diversity or singletons match your observed data. If they're close enough, you accept those values for demography. You rerun this, that generates a posterior distribution for possible demographies. You can then use that to basically ask, under those demographies, how outliers are the values you actually see. So I'm basically trying to generate a more neutral, more natural, non-equilibrium null model that otherwise fits the data. Of course, the issue here is, do you simulate over the right structures? You can also use the empirical site frequency spectrum. A lot of these methods assume the Waterson, that's that distribution that's under equilibrium neutrality, and we ask how that's perturbed. Well, if instead you simply take punitive random markers and use those and look at the site frequency spectrum in those markers, that then gives you an example to compare your particular aspects to. So these are all, I'll call these pseudo-statistics. They're ad hoc, but they're in a reasonable direction to try to improve. The problem is you simply can't put p-values on this. This is basically about allelic surfing. Allelic surfing does not affect all genomic locations equally. It's especially troublesome since humans, Melanogaster and Simulans of Arabidopsis have all undergone range expansions, so those are often looked at for selection. They could feel the brunt of this allelic surfing. The last question, which isn't talked about quite as much, is there's been a trend recently to ask, is the appropriate null model the equilibrium neutral model? And the answer is probably no. Even if you can be assumed that equilibrium works, the correct null model is probably background selection, because we know it's occurring. Background selection gives you a different structure than the neutral model. So an equilibrium background selection gives you a different model than the equilibrium neutral selection. So in particular, if I'm doing, let's say, an empirical site frequency spectrum, I've got my gene over here. I'm trying to ask if its site frequency spectrum is unusual relative to a sample of other site frequency spectrum from the population. In a neutral case, I simply pick a random set of those and use that for comparison. In the background selection case, I'd have to also adjust and pick only markers over here whose genes are in a comparable gene density to recombination ratio part of the chromosome. Why? This is what sets the strength of background selection. If gene density is rich relative to recombination, I should have stronger background selection. Why? Increased gene density, more deleterious mutations. So I want to then, if I'm comparing this empirical site frequency spectrum, I want to then go ahead from my neutral sample and only pick site frequency spectrums that are from comparable regions where that chromosome has a gene density to recombination ratio relative to mine. That's how you use background selection, and a lot of people are now thinking that's a more appropriate, and I agree, a more appropriate null model than neutrality. So it's still not getting at equilibrium, but it says that we need to move away from neutral equilibrium to move away from background selection equilibrium as our default starting model. Yeah, sure. Yeah, so, and you can also modify that from away from equilibrium as well, but what we're doing here is we're replacing the double assumption of equilibrium neutrality with the assumption of equilibrium background selection, which is a much richer and therefore more complicated and harder to model class. The nice thing about neutrality is you model with the mutation rate times the population size. You're done. Background selection is a bit more tricky to modify, but the nice thing is with genomic data, you can use genomic data to make inferences about what the background selection patterns should be. Okay. So let's look at allele frequency change. This is the most logical test, right? I sample a population, maybe do a selection experiment, come back later. I look for alleles that have changed excessively. Excessively means changed more than I expect from drift, or conversely, since drift causes allele frequencies to change, alleles that change less than I expect from drift. There are power issues, especially on less than you expect from drift. The time scale that selection changes allele frequency is 1 over S. The time scale for changes in drift is 1 over 2n. So what you basically need is your data point times your sample size to be much bigger than S. So your time span times your sample size. If you don't have that, then you run into power issues. So let's look at the simplest way to look at these. I have basically an allele frequency at time t, allele frequency at time 0. In the early literature, people simply ask whether the two allele frequencies were significantly different from each other. Seems pretty straightforward. The problem is it doesn't acknowledge the fact that there is an evolutionary variance that happens, and that variance depends upon the strength of drift. So here is the variance in that allele frequency. Its expected value is P0 in the drift, but there's the variance in there, and the variance increases as with time, so increases with drift. We can basically take the divergence as the new value minus the old value, and we can then construct a test here. And this basically is the variance of that initial frequency. Here is the variance at time t, So if you add those two variances, you get this expression over here. So the idea is, if I'm doing a t-like test, I have a difference divided by a variance. And that variance has to account for the fact that it's a function of the divergence time. If I just simply compare allele frequencies and ask if they're different, I'm not accounting for that divergence time, and therefore I'm not doing a proper accounting for drift. So again, chapter 9 goes into this detail. And here's basically the test statistic. It's basically a square difference divided by a variance. That's basically a chi-square, because if I look at the difference divided by the square root, that's a t distribution. Remember, a t with 1 degree of freedom, if you square it, is a chi-square. So these are expressed in terms of chi-squares, and this is the Waples adjusted test. And the Waples adjusted test basically is to ask if this is different, but wait, we're not there yet, as they say in the info commercials. Wait, there's more. What do you take as P0? Well, I've got a sample at time 0 and a sample at, say, generation 15. You might say, oh, P0 is simply your sample point at time 0. But you know that the sample point at time 15 also has information about P0. So you combine those two and get a better estimate, and that gets us into the general linear model. So, time to take a break here. How many of you have had a reasonably good stat class? How many of you have heard about the general linear model in your reasonably good stat class? So the idea is, most of what we use in statistics, you can write in a framework of something called the general linear model. Is the light good enough to see over here? Yes, no, eh? Okay, I'll put the light on and see if that helps. And the problem is, I want to write here, but I, sort of can't. We can go to the blackboard and back, but then everyone will be dissatisfied, and I'll be off camera. Well, yeah, but it takes like 5 minutes to raise and lower the screen. That's the problem. So it's a good way to run out the class. So the general linear model basically says this. A huge number of statistical models I can write as y equals x beta plus an error. Beta is the unknown, and beta equals x transpose x inverse x transpose y. That's assuming the errors are what's called ordinary least squares. If I have general least squares, it's this. So we have a method that's basically based upon a matrix of information, X, a matrix of observations. So for example, that general linear model is a t-test. That's analysis of variance. That's a multiple regression. That's analysis of covariance. So most basic linear statistical models you know of can be written in general linear model form. And so the idea is I can borrow information from the current value of PT to estimate P0. And to do that, I need to know a couple things. I need to know the variances of those. Here's the sample variance of P0 and PT, I know what that is. Here's the sample covariance, and I've got expressions on the previous page for that. Then basically, this is an estimate here. That's a number 1 by 1 divided by a number. This then basically uses the information. P is a vector here of the frequency of time 0, frequency of time t. This gives me an improved estimate of P by taking the original value of P, the value of P at time t, realizing there's a covariance between those, and using all that information, and basically here's the expression then that basically updates it. That's the expression from the general linear model. So there's the general least squares estimate of that. And what it does is, it, instead of taking P0 as a value from the initial sample, it takes the initial sample value, the current sample value, and basically adjusts for the fact that they're correlated and they contain information about each other. So basically, it extracts all the information that's in the sample out to do that. And so you get that expression. And this then basically is what's called Waples adjusted test, is to adjust for this. Let's look at an application of that. Here we go. Hey, Lepidoptera, I'm excited. So this is a very classic study by R.A. Fisher and E.B. Ford, and they're looking at this scarlet tiger moth. And this scarlet tiger moth has 3 color forms. You'll notice the absence of this discal spot here and the absence of the cells here. It turns out this is a single locus, homozygote, heterozygote, homozygote. Pretty cool. So this phenotype here is homozygous for this allele. This phenotype here is homozygous to the alternate allele, and that's a heterozygote. So the cool thing is you can simply capture these things and count the allele frequencies. Pretty cool, right? And there's a little fen or marsh outside of, I think, Oxford. Yeah, outside of Oxford. Cotton Hill Fen, right? Sounds like a, one of those really sappy English romance movies. Have a lonely lepidopterist out there or something, counting the tiger moths. So what they did was they have a large series of data. In fact, this is a really complete data set from 1941 to 1979, and from 83 to the present. It's a huge data set. What they observed was the frequency of this allele went from about 9% to about 4% in an 8-year period. They have the effective population sizes here. What you can basically then do is for this 2 sample, you can first of all figure out that's the variance from the data here about sample sizes. There's the sample sizes and effective population sizes. There's that covariance structure. You can then estimate that. So here is your best estimate of the initial frequency of that allele. It's 7%. You can then compute the sampling variance by plugging it in, and getting that is your variance of that square difference, and you compute the test statistic, and there it is. This is a chi-square value of close to 7. The value probably of a chi-square is bigger than that, is less than 1%. That implies a strong rejection of neutrality. Now that rejection arose because I made this assumption the effective population size is 1,000. I know time is good, so I can then go back and try different values of the effective population size and see what effect that has. That's just simply redoing the calculation, and this now would be 2 times the current effective population size, same thing down here. So if I go ahead and do that, taking the effective population size at 500, that test statistics becomes 4. It's still significant. If I take the effective population size at 250, it's no longer significant. So an effective population size slightly smaller than 500 would still be compatible with drift. So one nice thing about this Waples adjusted 2-point test is I can try different effective population sizes and sort of find that critical population size above which the amount of divergence is not consistent with drift. And then you have to ask, as a biologist, is an effective population size of 500 reasonable given what you know about the biology, or given some other independent estimate? So this is a simple test based upon looking at allele frequency change at 2 time points. Tomorrow, we'll take this exact same data and do the Fisher-Ford test, where it's not 2 time points, but it's any number of time points with any amount of difference between them, and that gives you the sort of more general form that we'll talk about. Okay? So we're talking about tests of selection, and after an overview of general methods, we're now starting to drill down. And I'm in the middle of talking about the kind of classical test, which is you measure allele frequencies over 1 or more time points. And we talked about the 2 time point test, the Waples adjusted test, where you basically measure a allele frequency and measure it later. And the key idea there is to estimate the initial frequency, you use the initial frequency and also information from your second time point, because those are correlated, and you do that in the framework of a general linear model. Now, after questions, we're going to talk about the more general question. This is called the Fisher-Ford test, where instead of looking at 2 time points, you have a series of time points. They don't necessarily have to be the same distance apart, and so we'll talk about that method in a second. But before I do, as always, we've covered a bunch of stuff so far. Questions? So you have a couple empty seats this morning, a couple of slackers. You'll have to tell them about the Power Window video. So we've covered a whole bunch of stuff. I posted the linear model notes as well. Just, I mean, not formal notes, but unofficial notes. Did people see those? Or some people? Oh, I know who's missing. 2 people who have the copy of the book. Yeah. You're still eating. See. You bring your food in here and make the room messy. That's what you do. Okay. So in the Fisher-Ford test, the way they proceed on this is slightly different. And the first thing they do is they take the frequency data and they do this very famous square root, arc sine of the square root transform. Now, Fisher basically showed that when you do this transform, it basically makes the frequency data homoskedastic. Remember the term homo and heteroskedastic? Homoskedastic means that all your data have the same error variance. That's not the case with allele frequencies. With allele frequencies, the error variance is P1 minus P, so it varies with the frequencies. A classic way to remove that is to use not the frequencies themselves, but the arc sine of the square root. And basically, Fisher showed when you do that transform, the data are now normally distributed with the initial mean, which is basically the transform mean, and then this drift term here, because this has a constant variance, this drift term here depends upon T and E. So the idea then is you have a vector or a list of your allele frequencies that you observe, and you transform those allele frequencies, and so now your vector Y of information is basically this, a vector of the transform allele frequencies. Then you have a vector of 1, and what you can basically show is for this model, the variance for data point I is the sample variance and the evolutionary variance. Sample variance has to do with sample size. That is, I'm trying to estimate this frequency from a finite sample. The second variance is the evolutionary variance. It has to do with the fact that the true mean is going to be, so this is basically the error in estimating the true allele frequency from a sample. This is the fact that the true allele frequency itself is changing over time, and that's the variance in that associated with drift. So you basically put all those together, and you can get an initial estimate for the original frequency. And remember, we see things like this, V inverse. So basically what this does here is it takes this data and it standardizes it to have a common mean, because remember here the variance differs over the observations. A common mean and the observations are uncorrelated. Then you can basically generate a deviation. So Y is this vector of transformed allele frequencies, and this is basically your estimate, your updated estimate of the allele frequency transformed at time 0. This is a vector of deviations. So the first observation would be the allele frequency from observation 1 minus so allele frequency at time 0 from its expected value, allele frequency at your second data point from its observed value, down to allele frequency at your last data point from your observed value. And the key is the distance between data points don't have to be the same. They can be anything you want. You just have to know what the generations are. Then you basically have a vector then of deviations. Then this is your test statistic. It's basically the vector of deviations times V inverse, that is the sampling variance for these, times that. So when you basically do this, it takes these data and it makes these data the sum of a bunch of squared unit normals. Remember, you see something like a quadratic product with an inverse, what that does is it takes data and it basically makes the data independent, and it makes it all have the same variance. And in fact, in this case, it makes it have independent with variance 1. So basically, you get a sum here of k minus 1 terms, and that then follows a chi-square distribution. So let's go through an example of the Fisher-Ford test. Again, there's lots of detail here, and my purpose isn't to take you through half an hour line by line in each of the steps. You can look those up and follow it, follow the example in the book more clearly. The idea is the basic approach. The basic approach is you've got a series of allele frequencies. You transform those to make those better behaved. Then you basically estimate the initial frequency. Then you compute basically a vector of deviations, which is weighted by the sampling error. So here are observations for, and what we're going back for is this very famous data set on these 3 genotypes that give you different wing characters in this very attractive tiger moth. It's called the scarlet tiger moth. And so, actually, let me just, it's always fun to throw biology in here because I know we're all biologists. So tiger moths are very colorful because a lot of times they sequester really nasty stuff. Like a lot of, for example, feed on milkweed and are pretty nasty. But tiger moths do something interesting as well. So these are brightly colored because they're day flyers. That makes sense, a brightly colored day flyer. You want to tell a bird you're distasteful. When do most moths fly? At night. What's the major predator at night? Bats. How do you tell a bat, look how colorful I am? What's the expression? Blind as a bat. So what the tiger moths do is they send out an acoustic signal. So they're acoustically aposematic. Instead of giving you a bright warning color visually, they give you an acoustic signal to tell the bats, go ahead and eat me if you want, but you'll be sick. And so there's really a lot of interesting biology based on that. They've got an ear on the tip of their abdomen, and they use some of that acoustic signaling for mate choice, et cetera. So that's some kind of random background biology. So basically their estimated frequencies over 4 years. Here's the sample sizes, there's the transform data, there's the variance covariance matrix, here's your vector of observations, that when you take this data and these observations, your updated initial frequency turns out to not be 0.62 but 0.54. Again, that's borrowing information from these other observations, because the frequencies here are correlated with an initial frequency, and you can borrow that observation and use that. And when you go ahead and compute the vector of deviations, it's 7.8. Compare that with a chi-square 2, you get basically a p-value of about 0.2. That's assuming here, I slipped that in there, that's assuming an effective population size of 1,000. So under effective population size of 1,000, this observed change in allele frequency is too excessive. Well, what you can also do is try something else. If you try an effective population size of 500, now the test statistic becomes about 5.4, and now the p-value becomes non-significant. So if your effective population size is basically 1,000 or bigger, it's pretty clear that the data are not consistent with drift. If your effective population size is 500 or less, the data are entirely consistent with drift. So you're in a bit of a quandary. What's the effective population size? You may see thousands of moths flying around, but as we saw in the effective population size, there are lots of ways that the effective population size can be far lower than the census size. So these tests for allele frequency shifts, they're often phrased not in terms of a p-value, but in terms of what's the critical effective population size above which the data are not consistent with drift. And there's a huge category of tests that follow along this. For example, in the SARS outbreak happened a couple of years ago, Fisher-Ford was dusted off, and a number of people basically came up with Bayesian modifications of this basic idea. So that's kind of a summary of allele frequency shift tests. One more modification, and that's Schafer's linear trend test. And under the simple drift, the model is mu plus e. It is the allele frequencies are the initial frequencies plus an error, because you're randomly diverging about that initial frequency, but your expected value is 0. That's why you can use those those other frequencies to estimate your initial value. Schafer said, well, we can make this test a little bit better. We can do Fisher-Ford, then we can also ask, is there a linear trend? That is there a trend in terms of the allele frequency changing? Because if it's just changing randomly, well, that could be drift. But if there's a linear trend on that, that's further additional support. So you can basically fit this model here. Now your x and beta matrix in the linear model becomes a vector of 1, and now a vector of time point, and your second unknown is this slope here. You can basically then test and ask, is that slope significantly different from 0? And if so, it's further support, because not only do you show, number one, the deviations are too large relative to drift. To do that, you basically run the Fisher-Ford test, so you basically not have beta, you'd fit a first model. Then you'd fit this second model and show that that linear trend term was significantly different from 0. So it's just another bit of information of support that suggests that your data, the allele frequencies, are not random, because number one, the divergence is too large, and number two, if the slope is significant, that not only the too large, but this is also a linear trend, and you'd expect a linear trend under selection in allele frequencies. You wouldn't expect a linear trend under drift. Okay? So those are kind of introductions to looking at allele frequency changes. The problem is you need to basically have enough time points relative to the strength of the allele frequency change. And furthermore, these are applied on a locus by locus basis. And usually the way people apply these is they have some obvious candidate gene. Beta hemoglobin for sickle cell anemia, this pigmentation gene, maybe a disease-resistant gene, or an insecticide-resistant gene, and they follow it over time. So these things aren't really you can scale these for scans, but they're not really amenable to genome scan. Remember, in a genome scan, you basically make no assumptions about what the potential targets are. You just scan every site. Okay. So questions about these divergence in allele frequencies over time tests. They're kind of the most sort of straightforward. So the second category of divergent tests is I can have allele frequencies diverging over time. I can have allele frequencies diverging over space. I've got a central population. I have daughter populations that come from that. I can then ask, is the divergence among those daughter populations excessive? And so these are between population divergent tests. And we gave an example of dairy versus meat breeds, but more generally, suppose I've got several populations. And the way these tests proceed is they use FST. FST is a general measure for how much divergence you see. If I take a random population in nature, I expect some FST because that population has some sort of structure. There's some subpopulations that have migration between them. So the way the FST tests work is you have a large number of markers. You test all of those. You compute an average FST. Then you test individual markers and ask if individual markers have an FST that's too large or too small relative to the population-wide sample. That's the basic idea behind these. Now, there are modifications we'll talk about as we go through that. Yeah, and what this basically points out here is the two population tests, you can basically do like a two-point time series test. So for more than two populations, we use FST, and this study of looking at patterns of genetic variation over space often has the fancy name of landscape genetics, right? It tells you the importance of packaging. Because someone says what you work on, you can say, I'm a landscape geneticist. And the public will think you kind of work on fruit trees in their backyard. So by the way, I do a fair bit of traveling. And when you travel, sometimes you sit down next to someone, and they're pretty interesting, so you want to chat for a while. And sometimes, my God, it's like, I hope this person doesn't talk to me. So my strategy is one of the initial questions they always ask is, so what do you do? So since I'm a statistical geneticist, if I want to talk to them, I say, I'm a geneticist. Then I hear 8 hours about how their aunt had green or blue eyes, and does that mean, are they related? Or if I don't talk to them, I say I'm a statistician. And it's like crickets, and I have a nice, comfortable flight. So you're welcome to try that trick on your own at some point. So let me remind you about FST. So FST is simply a way of looking at the amount of divergence. So I sample a series of subpopulations. So I have to specify those subpopulations and sample allele frequencies in those subpopulations. Then I compute an overall allele frequency, and I compute a frequency of between deme variation. And FST is simply the fraction of overall genetic variation that's due to differences in allele frequencies across demes. If you simply have drift acting, what you can show basically is FST over time is simply going to be a function of drift. And that's because you take your initial variation and you're partitioning it through drift in here. And this is often why it's called a fixation index, is because FST goes to 1 as time gets large enough, because the demes get fixed for differential alleles. In more complicated structures, so for example, here's an island model. In an island model, everybody routinely exchanges migration with everyone else. You can have other models, though. For example, hierarchical models. Here you've got two demes that exchange at a fairly high rate, same fairly high rate, and they exchange between at a fairly low rate. You can also have what are called isolation by distance models. If we're populations spread out in space, the closer you are, the more frequent migration is. And the key feature is all these different types of migration structure give you different levels of covariances, that is, correlations among different demes. In an island model, there's no correlation because everyone exchanges equally. In a hierarchical model, these will be correlated, those will be correlated, and these, between these groups will be lessly correlated. So all of those things will end up mattering in terms of looking at what FST is supposed to look like. And that's why there's so many of these FST-based tests. So let's talk about whether FST works or not. And here's a nice little example. This is the question? Yeah, sorry. Between, sorry, deems? Great question. The answer is, well, so they're the same thing. I mean, in a sense, people can talk about a population as a set of deems. So usually deems are referred to as subpopulations. So think about, this room is the entire population, right? A deem could be this first row and this second row. So you'd be a subpopulation. People, that's being really technical and anal, which, you know, like that great question, is there a hyphen in anal retentive? Question mark. So we could view this first deem as our population we're sampling and call it a population. But usually when you hear the term deem, we have a set of populations we're sampling, so we can talk about a metapopulation for everything. So there's several different ways. You can talk about population being a deem, and a metapopulation is a collection of all those deems. You can talk about a population being all the deems, and each deem is a subpopulation. That terminology goes back and forth. So population, yeah.
Just for clarification, is this due to sampling or to a structure?
No. So the idea, that's a really critical question. What you're assuming when you specify a deem or a subpopulation, that is your sampling unit, and you're assuming that you're measuring a cohesive group of individuals that may be exchanging genetic material amongst themselves at a much higher rate. That is basically when I take a deem, I'm assuming it's a random mating population. And then there's some other population that it may be exchanging to, but it's not necessarily part of your random mating population. So subpopulations and deems are a sampling scheme. So if I have populations, say, along the seashore, there's no natural clean deems. It's a continuous population. But I could sample in Southern California, Central California, Northern California, Oregon, and call those deems and ask, are those populations different for, what's the FST for those populations? When in fact it's a continuous smooth gradient, right? So the key to population structure is we have kind of simple models of kind of discrete units, but in reality, population structure is kind of like a color-coded heat map. We've got, densities of populations and lower densities, and they're all kind of smoothly connected. But we'll show how to get around that when we talk about the third generation tests. Okay. So here is the tobacco budworm. There's the little larvae, very cute. And so Martin Taylor, who was at Chicago at the time, was basically looking at voltage-gated sodium channel genes. And the reason those are these pyrethroids, basically insecticides made from chrysanthemums. These pyrethroids basically attack those. And so what they were basically asking is, well, is there excess divergence in these sodium voltage-gated sodium channel genes if I look across populations of this? Because certain populations have been exposed to pesticides and others haven't, just given regional uses. What they basically showed was they looked at a wide sample across the US, and the FST value for the HPY gene, the gene in the sodium channel, the FST was about 4% plus or minus 0.5%. Then they took 14 other random loci, and those were 0.2% plus or minus 0.1%. So there's much more structure in this gene than there are in a random background set of genes. So it shows much more divergence among populations, and that makes sense if you have different amounts of pesticide usage historically, and in fact, the populations were chosen for that reason. So this is kind of an example that basically shows you the underlying idea behind FST. And so FST then says, okay, I have a whole bunch of candidate genes, markers, and I scan them one at a time and compute the FST for that and ask if it's excessive relative to some standard. Right? That's the basic approach. The problem, of course, is you're always going to have values which are outside the mean. So what is excessive? And so usually to get around that, you have some sort of idea what the distribution is. As we'll talk about in a second, the early ideas was that distribution of F values was a chi-square whose scaling parameters were set by the overall average. As we'll see, that's not true because that basically assumes a specific model, the island model, which basically makes observations between populations uncorrelated. When you put correlations in, you change that distribution. So the early way people got around that, and you still see today, is the so-called outlier approach. Here's the FST value. Hey, look at this value way out here. That's got to be significant. Well, it could be chance. But outlier values are kind of cheap and dirty. You do your test statistic, whatever it is, and you get a nice red pen and you circle all those in the tail and go, those have got to be interesting. Not necessarily. It's not a formal statistical method because you don't know a p-value unless you know something about the distribution of those. At a minimum, the variance in that. But outlier methods are often used, and they're often called enrichment methods. So you got a whole bunch of markers, you find the outliers, you circle them, then you go back and try to ask more biology about those. So a lot of the approaches for declaring selection are very heuristic, and they simply say that these values are outliers. Now, if you have selection, selection is going to generate outliers, but just because you have an outlier point doesn't mean that locus outlier was generated by selection. And that's kind of what this says, blah, 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 blah. Right? Blah, 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 blah. So one really interesting example was that there's this nice study here that basically found that river fishes have an extraordinarily large number of outlier loci. So of course you go, man, there must be something really interesting about the ecology and biology of river fishes. Maybe some selection because of where they are on the river, or maybe it's the fact that that's a linear migration structure that gives you a fractal structure, and that fractal structure then gives you a very high variance in FST, and that's what's generating the outliers. Right? So remember, FST is a number, but underlying that number is this very elaborate process about how subpopulations and demes exchange with each other, how correlated they are, and the early models basically assume that they're all uncorrelated with a constant variance. That gives you a very different distribution than if they're highly correlated. And so one of the things we'll talk about at length about is the new versions of FST basically try to get at the correlation structure among your sampled populations to adjust for that. So one issue you often see, if you go back to what FST is, there's FST. If you take the log of FST, you basically, so log of 1 minus FST, you basically get this expression here. So what this basically shows is you can take FST and essentially turn it into a amount of divergence. And these times are often called branch lengths. And this basically gives you something called the population branch statistic, or PBS. I think someone here mentioned they're working on population branch statistics. No? Okay, so, and here's the classic example. So you take a whole bunch of genes in Tibetan, in Han, that is the majority of Chinese, and of course the Danes, just because they're Danish. And what you see is that the divergence for most genes look like this. If I take an FST and look at pairwise FSTs, and then do this transform and transform those into distances and put a tree together, what you basically see is that the Tibetan and Han Chinese are rather similar and somewhat disconnected from the European Danish. If you then go to, for example, EPA S1, a gene that's thought to be involved in adaptation to high elevations, what you see is this tree is blown up, so the gene evolves a bit faster. What you see here is that these equal branches here, you have an enormous branch here, so in Tibet there's an excessive amount of divergence. So that's one way of kind of looking at FST, is to take FST distances and turn them in essentially into distances on a tree, showing that there's excessive divergence in this gene relative to the divergence you see here. Notice here one thing though, that this here is like a shrunken down version of this. So for any given gene, the actual size of here can vary. What you want to look at is that when you scale it appropriately, if you scale this appropriately, that branch should be this long, not that long. So that's one general way of using FST. It's nice graphically because you can make a big cases like, well, clearly there's selection on that. But what you don't see is if people did that on several hundred genes, what these trees would look like. And so just because you see outliers random processes are always going to generate outliers, right? Always. That's why, for example, underdog teams win. They won't win on average, but it's a random statistical process, and outliers do occur. So you have to be very careful when you are looking at data to not get overly excited about your outliers. If before you do an experiment, you expect a particular gene to be an outlier for reasons that are independent of the method you're testing, and that gene does turn out to be an outlier, you're okay to get excited. But if you're randomly scanning genes and see an outlier, that's an ascertainment issue. Because you know some values, if I just randomly generate numbers from a 0-1 uniform distribution, I'm going to get some outliers. There's nothing special about those outliers, it's just random chance it generates them. So just seeing outliers by itself isn't enough. And so if you see a figure like this, you basically have to dive into the paper and ask if this was a gene they were interested in beforehand, or if they just sampled a whole bunch of genes, this was the most extreme one they saw. If they sampled a whole bunch of genes, and this is the most extreme one they saw, it doesn't mean that it's not a good evidence for selection, but it basically means you've got to be a bit more careful. If they said we're going to test this gene beforehand and then gather the data, this would be a different level of support. So you always have this issue of if you sample a whole bunch of markers and then pick the largest, that's a weaker signal than having a particular candidate without knowledge of any of the tests you're using, and then showing that candidate gives an excessive value. That's a much stronger level of support. Yeah, question?
So then is there sort of like very complicated to understand support for genome-wide?
Well, we'll talk about how you do that for genome-wide. I'm just giving you kind of some background, you know. So the key points so far are the notion of population structure measured by FST, the notion that we can take an FST value and turn it into a time or a branch length. We can ask if some divergences are excessive, that is, the time to get those divergences under drift is much larger than we expect. So you have an excessive amount of divergence relative to the time point, right? Those are kind of the key backgrounds. And then the idea that if I sample FST over a whole bunch of markers, I can compare specific values to some mean average and then try to use that to ask if certain ones are excessive or not. I haven't told you how to do that, but that's the basic idea. Those are the key points so far. Okay. Questions or clarifications on those? By the way, for those sitting in the back, there's 3 lovely seats over here. But I know you like the tables back up there. So. Okay. Well, we should maybe put like a big boy lounger in there. You know, you just kind of kick back and have a drink holder and you'll be all set. So the key person that got these FST tests really interesting is Dick Lewontin. Dick Lewontin is one of the more famous population geneticists. He has a very nice kind of chatty book that came out. It's fun to read back in the 70s from, I can't remember the exact title, from Columbia University Press. It's basically, Lewontin was the one that got molecular population genetics going because he and Hubby, who was a chemist, showed that you could basically look at protein variation by using starch gel electrophoresis, and all of a sudden you could move from looking at phenotypic variation to looking at molecular variation. And this was pre-DNA. And so they found excessive amounts, and basically that excessive amount of molecular variation from looking at protein variants has now carried over into whole genome sequencing. And Lewontin was one of the people behind it. These are his famous equations here for two-locus selection. So the paper was Lewontin and Krakauer. What a great name, Krakauer. Sounds very volcanic, doesn't it? So in 1973, they had a very famous paper that came out that showed that if you have an island model, and an island model means all the populations you sampled exchange genes equally with all other subpopulations. That means there's no correlation structure. No 2 demes are more similar than any other 2 demes. And that only happens under this island model. He showed in that case, the F statistics follow a roughly chi-square distribution. And we used this idea when we talked about QST being roughly chi-square once you scale it as well. These tests were pretty quickly shot down because people realized that if you don't have an island model, you don't have a chi-square distribution. And so there was second generation tests we'll talk about, which says, okay, let's assume a hierarchical model. Now what's FST supposed to be? So second generation tried to make the tests a bit more model-based. We'll talk about those a little bit. And third generation tests, the ones we have now, basically make no assumption about the model. They just simply take the data and use the data to construct a covariance matrix of how deems covary given the marker information. And these are probably the most accurate, and these actually make FST statistics sort of make sense. So let's kind of go through the logic there. So Lewontin-Krakauer basically says that if I take my FST value, my random variable, and multiply it by this, 1 minus the number of deems over the average value, that's roughly chi-square with n minus 1 degrees of freedom. So they got this by assuming that the deems are uncorrelated, and when you can do that, you can get this chi-square structure. One immediate problem that you can correct it with is the bar here means I'm taking the average FST over everyone. So each of you is a different marker. I compute this FST bar by taking each of your FST values and taking the average. Well, that sounds reasonable, but it's not reasonable in the sense that suppose this row here, these are all loci under selection. They're going to have extreme values. They're going to inflate this over what it should be. Now, you expect that most loci probably aren't under diversifying selection. So how do you adjust for that? And a real standard way to adjust for it, it's a standard approach that you use in a lot of things where you get outliers, is not to take the mean, that is the average value, but take the median, the 50% value. So now what I do is I take the FST values for everyone here, and I compute the value, line them from largest to smallest, and take the value exactly at a half, right here. And what you can then do is you can make this scaling factor here. You can basically take the median value you observe and relate that to the median value you expect for a chi-square within minus 1 degrees of freedom. So for example, if I sample 5 populations, I have a chi-square 4, and the value for a chi-square 4 that 50% of the probability is below, 50% above is about 3.3. If I actually observe the median FST is 1.27, I look at that ratio and I now get this new inflation factor here. So these medium approaches are nice because they don't care how big these values are. The medium approach doesn't weigh how large those values are. The mean does, the medium doesn't. The medium simply just ranks them and takes the one in the middle. So when you have a lot of tests where you expect there to be some outliers which are really large because they depart from the null, a good, more robust estimator, if you want to adjust for something, is to use a medium rather than a mean. So that's one modification you can make on the Lewontin-Krakauer test. But again, the big problem with Lewontin-Krakauer is that they assume that allele frequencies are independent among deems, that is, there's no correlation structure. We've seen lots of examples where you could have correlation structure. Populations on a linear climb, as your populations which are closer have more correlated allele frequencies. So Lewontin-Krakauer fell out of favor because the covariance structure you expect isn't independence among deems, it's something else. And that something else changes your chi-square distribution, alters its degrees of freedom, and no longer makes this test appropriate. So one interesting recent suggestion to get around that from Mike Whitlark's lab is this cute idea called Outflank. Very, very clever. And basically, here's the idea. It's again based upon outliers. And so the idea is, here is what a chi-square typically looks like. And that should be F bar ST. And what they said is, well, you know, if you have all these covariant structures, you're going to depart from that chi-square on the tails. But if you look at the middle of this distribution, so 5%, 95%, or some other region you want to look at, you look at the middle of that distribution, it's going to be better behaved. And what you can then do is take your data in the middle and then do a goodness of fit and figure out what chi-square distribution it fits to. So for example, you may have 8 deems. If you have correlated structure, you really only have maybe a chi-square that has 3 degrees of freedom. So they suggest you basically truncate at both ends and then fit the middle of the data to a chi-square. A better way to do that is there's something called the gamma distribution. A gamma is a very general distribution that chi-square is a special case of, where one of the parameters is discrete. Gamma allows you to basically have non-discrete, so basically if you've got fractional degrees of freedom, the gamma will basically fit it well. So the idea about Outflank is what you basically do is you take your data, you throw out the lower and upper tails, how much you can fiddle around with, then take that middle part, fit a distribution to that. With that new distribution, then you can look at what the p-values are in your tails. So it's kind of a way to get around a little bit of these limitations of Lewontin-Krakauer, and their simulations showed that it worked pretty well. So that's still using the Lewontin-Krakauer idea, but a very simple modification of it. So the first generation Lewontin-Krakauer test, you take FST, either look at outliers or you look at specific FST values, and you can put a p-value on them by fitting a chi-square distribution, or maybe by using an updated chi-square by Outflank, as we've talked about. So questions about those so far, before we move on to second and third generation tests. FST tests are really really popular because, you know, with landscape genetics, people are taking subpopulations and measuring genetic diversity in them all the time. It's the natural data they have. You expect excessive divergence in those if you have selection, so it's a very natural data set, and so people are very naturally drawn to using FST type values for that. So the second generation, the next wave of Lewontin-Krakauer LK tests, is to move away from the island model assumption to other assumptions. And one of the more interesting assumptions was Bayes FST, and Bayes scan is an example of that. And the idea here now is that instead of our populations all exchanging, what I have is I have a core population, and each deme is a random draw from that population to start it, and no further migration since then. Right? Whereas under the migration model, you're exchanging migrants every time. So you have a different population structure, and so the FST and Bayes scan programs assume that population structure, and then basically look and see how good you are. And there's other variants on that. The problem with second generation tests is they're an improvement because you're no longer assuming the island model. The problem, though, is you still are assuming some model. And that model may be more general than an island model, but it's not completely general. If you think about population structure, it's such a high dimensional space that you can have. There's no obvious canonical model for population structure. So second generation approaches were better because they move you out from this constraining island model, but they still have issues. Third generation approaches basically say, I take my marker data, I use the marker data over all the markers to compute covariances among all the demes. That's a very general approach, and I can use that very general approach then to then get a much better estimate of what my distribution is going to be. Right? So instead of assuming I've got no correlations or I have a specific number of correlations, you actually use the data to tell you what those correlations are. There's a whole bunch of tests. You probably have heard of some of these. There's the FLK test. There's the FLK test. There's Bayes environment and Bayes environment 2, PC adapt, other things like latent factor mixed model, LFMMA will come back in a second setting. So the idea behind all of these is you take your marker data and you use that to construct the covariance you expect among demes for those random markers. That gives you a better distribution. Then you see if that better distribution, then if your particular marker you're scoring is an outlier against that much better distribution. Here's what the kind of test statistic looks like. These are our old, look at our old friends here, right? Quadratic products. Vector, inverse variance covariance matrix vector. So this term here, F inverse, this is in the FLK, this F inverse here then is an estimate from other markers of your covariance structure among demes. And that then gives you a test statistic. That test statistic is chi-square distributed, and the nice thing here is this inverse matrix here cleans up that chi-square by properly accounting for all the correlations. Right? The Lewontin-Krakauer test doesn't do that. This actually cleans it up. There's a bunch of variants off this. So those are third generation tests. I collect data, I use the marker data from some carefully chosen set, and the issue, of course, is how do you carefully choose? An easy way is simply to randomly sample from that and compute F matrices and look how stable they are. You certainly don't want the marker you're testing, you don't want that marker you're testing to be in your estimate of F. In fact, a good way to go is to basically make an estimate of F, test each marker, and then take those markers which seem excessive, throw those out, recompute F based on the other markers, and run the model again. So those are third generation Lewontin-Krakauer tests, and they basically, again, are looking at FST, and what they do is they can now put a p-value on whether something is an outlier or not. There's still problems because you're assuming the markers you're using to estimate your covariance matrix among the demes, you're assuming those are honest markers. These aren't markers under stabilizing selection, and therefore will have too little divergence. They aren't markers under divergence selection, therefore will have too much. And so that issue aside, these third generation tests are probably the most powerful way to use FST data.
I guess I got a little confused because maybe from the previous classes that you had that you mentioned like using the neutral part of the genetic information to use the like critical argument and then try to test.
So the idea here is that when you make this F matrix, which talks about the expected correlations you expect among demes based upon population structure and migration, which you don't know, but you're making inferences about them based upon markers. You're assuming the markers used to make those inferences are neutral. If those markers are not neutral, you'll make improper inferences. How do you know they're neutral? You don't. So in a case like that, what you do is you just try different sets of markers and look at how stable your estimate of F is. The idea, if I'm looking at 10,000 markers and 1% are shaped by selection, that's probably going to have very little effect on F. If I'm looking at 20 markers and 5 of them are shaped by selection, it's probably going to have a fairly big influence on F. So there's always these little things you can do to subsample your data and look at how consistent your results are. That's kind of a clean way to do data analysis. It doesn't give you complete confidence, but it certainly gives you improved confidence. But the basic idea is you have a bunch of demes, you then compute from an appropriate set of markers the correlations among those demes, and then you use that then to adjust whether your things are outliers or not. So Lewontin-Krakauer, second generation, won't talk much about those, and then third generation tests. Third generation tests are much more robust because they make very few assumptions about the population structure. They let marker data tell you about what those assumptions are. So the questions so far? So the way people have modified these, and the idea comes from polygenic sweeps. If you have a polygenic sweep, allele frequency shifts are going to be tiny. If allele frequency shifts are going to be tiny, FST won't change very much. Why do you have allele frequency shifts? Maybe because you're responding to different environments. So an approach that is built upon these third generation tests is to look for if allele frequency at specific sites, if those change along with the environment. Now you have big problems in the sense that populations close together are going to share genes and shared environments. So you want to try to get around that. But this approach here is allele frequency correlations with environmental variables. It's often called environmental association analysis or genetic environmental association analysis. And basically the idea here is you add a factor in your model that basically asks if allele frequency shifts track with the environment. It's kind of like a Schafer trend test, and you're basically asking not simply if the departure is greater than you expect the divergence, but also if it correlates with some other variable. The initial papers were Anne Hancock, who did a series of papers where they looked in human traits in about 70 populations around the world. Then they took populations, for example, in high UV or different environmental conditions where the populations were scattered over the world, and there were ones nearby each other that had high UV, but there are also ones who were far apart, and then ask if there was a correlation between allele frequencies and UVs once you adjust for population structure. And what she found was there are a lot of cases where that actually does happen. So the basic idea, that one of the first examples of this, so this was human genetics, but it actually appeared in the ecological genetics before, was Joseph's spatial analysis, or SAM model. And SAM is really a simple idea. It simply says, I look for an allele frequency, and I map that allele frequency as a function of some environmental variable. So I basically have allele frequency, environmental variable. Then how do I map a frequency on a variable? That's a logistic regression. The idea behind these allele frequency and environment correlations, those go back to Dobzhansky. Dobzhansky was a famous geneticist who worked with Klein's and Drosophila. And he's maybe best known for a quote, which was, "Nothing in biology makes sense except in light of evolution." You've heard that quote before. And so what Dobzhansky noticed was, as you move up in elevations, certain chromosomal rearrangement frequencies shift. And they shift independently over multiple mountain ranges. So Dobzhansky had the best scam in the world. He was out of New York. He eventually moved to Davis many years later. And he was able to get a research system that allowed him to hang out in the national parks the entire summer by having his grad students sample bait buckets. So he basically sit in a nice lounge at Yellowstone and have his students run out and sample bait buckets and have them count the flies. Then over a nice meal, they'd give him the data and he'd make some grand interpretation and send them out again. But a student of his, Francisco Ayala, who's now at Irvine, took that scheme to another level. He said, why sample in the national parks when you can sample in the Caribbean? And since you're sampling in the Caribbean, you could rent a fishing boat, but it's much cheaper to rent a private yacht with a chef. And so go around these various places and send your grad students in a dinghy, have them row ashore, put out bait buckets, sit in the hot sun all day. And they were, of course, on the garbage scow that was falling. They wanted a nice yacht with the press sheets and stuff. So there's a lot of this idea about gene-environment correlations has a long history in evolutionary genetics. And the reason it's picked up a lot is people used to use chromosome rearrangements, very crude measure of genetic variation. Also interesting because a whole bunch of genes are packaged together at one and don't recombine. And then as people got more and more markers, this idea moved to that. And this spatial analysis method was one idea where you simply look at whether allele frequency is correlated with some environmental variable. Now the problem with that, of course, is you haven't decoupled the fact that when you sample adjacent populations, allele frequencies are going to be correlated because they're nearby, and also environmental variables will be correlated. So removing that fact that spatially close demes are going to likely share environmental factors and also going to likely share genes, that's where these Lewontin-Krakauer methods come in. So two of the most popular ones here are Koop's Bay and for Bayes environment and the latent factor mixed model. And basically these do the same thing. They just construct that covariance matrix in a little bit different way. A latent factor basically says that when I construct that covariance matrix, I do it one PC at a time, one principal component, and when I get to a certain number, I stop, whereas Koop constructs the whole thing directly, and there's back and forth about which one is better. The bottom line is they both kind of basically do the same thing. And here's the basic idea. The basic idea is, we've seen this before, vector, variance, covariance matrix, inverse, vector. This accounts for population structure, and this term here, this x beta, that basically gives you a vector. That x beta, these are adjusting for the environmental effects. You basically then ask, is beta significantly different from 0? And just like that linear trend test of Schafer, it's a second level of support that there's a selected factor that's driving these. Because not only do you get excessive divergence, but you also then ask if these beta are different from 0. That's kind of the grand overview. There's a lot of details we can drill down into, but the book covers those in gory detail. It gives you all the references. That's exactly what this takes care of. So, but your point is exactly correct. I mean, the issue with these allele frequency environment correlations are, let's do the following. Go to southern Arizona. Southern Arizona's got multiple mountain ranges. You go to the Chiricahua Mountains, and you notice as you go up in elevation that a certain chromosome rearrangement changes. Well, you know, that's a single observation. Those observations are correlated because the environments are similar, but you then go to another mountain range and see exactly the same pattern. Those are sufficiently separate that now that pattern becomes interesting. So how do you remove the correlations by nearby samples? That's essentially what takes care of that. That basically takes the fact that you expect things nearby to be correlated, and that adjusts for those. So when you see expressions basically vector transpose inverse vector, this inverse here basically is removing the correlations and giving things equal variance. That is, you're removing the correlated structure in the data. So it does that. It says once you remove that correlated structure in the data, do you still get a signal? So the test here basically is to ask, beta basically, think of that as a measure of an environmental effect. If beta is zero, it means there's no environmental trend. This accounts, it eliminates a term I don't like, in the sense, well, that's okay. What it does is it adjusts for it, right? Because it's always there, you can't remove it, so you want to adjust your data. And what adjust basically means is, you know, how you weight your data. So if two data points are really close, you know, you have to downweight that difference. If two data points are far, you upweight that difference. And this inverse here basically gives you the weighting you do on that. Right? So again, the idea about this is, if you're really interested in the details, the book goes through this in gory detail. If you want to kind of get the for want of a better word, the comic book view, which is still quite accurate, just basically think about the variance covariance matrix here, which you get from the Deems, adjust for population structure. You then ask, once you adjust for population structure, do you still see more divergence or more correlation with environmental values than you expect? Yeah, question? No? Huh? So this thing here, omega, is basically, that accounts for the expected correlation among Deems. No, not proximity at all. No, they basically, you measure your Deems, then you look at the correlations for random markers among those Deems. Right. So again, the nice thing, right, the nice thing about this is that you basically are given a matrix. You know nothing about if these things are close or far. So this is nice because it basically completely adjusts for that, and it uses the markers to adjust for that. So then it basically says, okay, sample 1 and sample 2, for a random marker not under selection and not correlated with the environment, how much do I expect them to covary? Then I ask, is my observed covariance excessive once I adjust for that fact? So this adjusts for that population structure, and this basically asks if there's an environmental trend. Yeah? You can put nonlinear associations in here by this. So if you have so x beta is basically a predicted value. So that could be, you know, environmental frequency equals A times some environmental factor, or it could be environmental frequency equals A times some environmental factor plus B times that environmental factor squared. So you can put polynomials in here very, very easily. Just like in a linear model, right? You can make a polynomial regression. This is basically a general regression, and that general regression doesn't have to be linear. The problem you have is when you start fitting nonlinear terms, you need more and more data, which is okay, but usually people will simply ask, is there a linear trend? Because even if you have a nonlinear trend, a linear trend captures that, and your concern here is usually not what's the nature of the trend. Your concern is there a trend? If you're worried about the nature, you can certainly make these much more elaborate. So I write this as x beta. That's just a general linear model regression. That can be as complicated as you want. It can be very nonlinear. Yeah, well, both. more populations and more, because if you also need more individuals. When this thing has multiple terms in it, you also have to worry about sampling error within a DEEM and between DEEM sampling error. So you want to basically increase sample size within a DEEM to reduce within DEEM sampling error and increase number of DEEMs to reduce between DEEM sampling error. So you can do it, but usually, people just ask, is there an environmental trend? And linearity often captures a significant component of that. If you have other reasons to suspect, you can certainly extend the models. I mean, models, you can always extend them ad nauseum. Sure. So, yeah, so if you have an Outflank, for example, put a quadratic term in there, you can ask, there's a quadratic component. That's a good point. The nice thing about these models is you can play with them as much as you want, right? But remember, the great statistician Box had a line that, all models are wrong, but some models are useful. He also had a great line, which was better an approximate answer to an exact question than an exact answer to an approximate question. So my philosophy for this course is I want to give you lots of tools. So when you have a problem, the way you attack that problem is driven by the problem, not driven by the tools. I think we all know labs that have one tool, and they take that tool and try to force a problem into that tool, and it's not a natural fit. The larger your toolbox is, we've given you lots of tools so far, the more you can look at a problem and then, address it. The analogy I always like to give is, you have a leaky toilet. If your toolbox is only a hammer, that's not very effective. If you do it right, you probably can. But if you walk in and the toilet's leaking, you can look and see, oh, I've got a wrench, I've got some duct tape. Versus if all you have is like a jackhammer, it's like, well, I can kind of fix the toilet by, kind of making a big hole in the ground and letting all the water go in there, right? So what you should do is the problem should dictate the tools, not the tools dictating the problem. And the more tools you have in your toolbox, the more you can be along that vein of letting the problem tell you what tools to use. That's always my philosophy in attacking problems in biology. Okay, so that kind of gives you, again, let me summarize. So we talked about excessive allele frequency change over time. We can take 2-time samples. We can take multiple time samples. 2-time samples, Wapel's adjusted test. Multiple time sample, Fisher-Ford. We can also ask, is there a linear trend? That's the Schafer's linear trend test. We then ask, okay, what about excessive divergence over space? That's FST. We talked about taking FST values and translating those into branch lengths. So you can look at what's the expected time if the divergence entirely drift and look for excessive branch lengths. We talked about Lewontin-Krakauer, and the key about Lewontin-Krakauer was you have a bunch of markers, you sample those markers, that gives you an average marker score. You can then use that average marker score to compute a chi-square distribution that is adjusted and ask if values are excessive. The problem with that approach is usually it's not the chi-square you think, because that Lewontin-Krakauer assumption assumed a specific model. There's no correlations among DEEMs. The Outflank method is one way to adjust that, to use your actual data to adjust your chi-square. But the most general way to go are these third-generation methods where you take your marker data, you let the marker data tell you what the covariance among the DEEMs should be, or is, not is. That makes no assumption about the model. Then you basically take that observed covariance structure and use that to correct your data. We can also look at environmental trends. We also can use that covariance structure then to adjust for the fact that adjacent populations may have correlated allele frequency changes. Okay? And that gets us through divergence in terms of time, divergence in terms of space. So questions? 2 really useful tools. FST type approaches are one of the most commonly used approaches in tests for selection, especially among ecologists, because it's the kind of data they naturally gather. So you pick up molecular ecology, that's basically a landscape genetics journal, and people are using FST tests all the time. The last approach I want to talk about in this lecture is I'm scanning now not a series of populations over space, but now I'm moving to looking at genomic data and I'm scanning along a chromosome. I'm looking for signatures of selection. An obvious signature is look at the pattern of variation. So nucleotide diversity, polymorphism, and the notion we've seen before is if I have a sweep, I should have a depressed region in that. Now I can also get a depressed region from differences in mutation rates. So what I want to then talk about here, the last part, are really briefly talk about visual scans, reviewing it, briefly talk about bottlenecks, maximum likelihood model. What these models basically do, and they first appeared, for example, for looking for domestication genes in maize, is when I look at maize versus teosinte, I should see a bottleneck. Across all my markers, I should see reduced variation because maize was a subsample of teosinte. Once I adjust for that bottleneck, do I see evidence that some genes have undergone a second bottleneck? So the first bottleneck would be the foundation, the subsetting for domestication. The second bottleneck would be selection on certain genes. So bottleneck models basically ask if the data are better fit, assuming that there's some fraction of loci that all loci have undergone one bottleneck, the common founding event, and that some loci have undergone an additional bottleneck. You can estimate that additional fraction. Those are bottleneck tests. Then we have formal tests you may have seen before, like composite likelihood ratio goodness of fit test, sweep finder, or cross-population composite likelihood ratio test. These are basically tests that take a specific pattern of variation and ask, is the curve of that pattern of variation what you expect if you shift a site frequency spectrum from selections? These are actually rather formal tests based upon maximum likelihood. So simple visual scans we see. Let me give you an example. So people like dogs for genetic models. And one reason they like dogs for genetic models is most of the shifts are caused by a few genes of large effect. So they're not really a good model for general evolution. But the nice thing is you can find things. So here we have two different breeds. We have the large Munsterlander, which I hadn't heard of before, but there it is. And then we have, of course, little dots in the infamous wiener dog. Key feature here is shortened limbs and coat color over here. So what people did is they took they had a candidate gene. So for the Mooselanders, the candidate gene was this pigmentation gene here. And what I'm doing is I'm going across a chromosome here, and I'm looking at each dot here is a microsatellite. Microsatellites were used fairly early on because they contain a lot of information. A microsatellite is a locus where you have a whole bunch of short repeated DNA elements, and because they're short repeated DNA elements, when replication occurs, they have high error rates. So the mutation rates in a microsatellite are on the order of 1 in 200 or 1 in 1,000, as compared to 1 in 10 billion for SNPs. So these loci then are defined by how many repeats. You're at 2, 4, 5, 6. And a 4 can easily mutate to a 3 or a 5 at very high rates. So they're highly polymorphic. So what they did here basically was they went ahead and they took these these dashed lines here are for the breed of interest. These over here are for a brown form and the dot for a control form. What you see basically is there's not a lot of difference between the three breeds. Then you get over here, and right around the candidate gene, what you see is reduced variation in the breed of interest, but not in the controls. You'll also notice, by the way, you don't have 0 at the target site. You've got this asymmetric thing. And that's not unusual. We talked about that. When you have a sweep, because of how recombinations occur early on randomly, it's not the case that right in the middle of the sweep is the target. That region of variation around that sweep is asymmetric. So if you tried to map the target gene, you'd say it's over here, when in fact there's a good idea that actually this is probably the target sweep. And one of the reasons you might see this is it might have been several different haplotypes, so you have a soft sweep, so you don't pick up a clean background. In Dotsons, This is a gene that's known to shorten limbs in humans, and so it's an obvious candidate. One thing you'll notice is interesting, though, is it's also a region of depressed variation in the controls, but not nearly as much as in the Dotson. So if you simply did the controls and did that, you go, look, there's selection going on here, but there's something else happening, maybe reduced variation, who knows. But this is why you always need to have a control. So these are other breeds, and what you see is in the Dotson you have this huge depression. Also notice it's asymmetric. There's the actual target. It's about 2/3 of the way through that asymmetry. So these are simple visual scans. In the early days, people used microsatellites because they had lots of variation. And if that microsatellite variation went down, it means that all the sample copies are from a relatively recent common ancestor. How can we take this idea then and make it much more formal? Well, recall that we showed in the lecture on population sweeps, the key sweep parameter is this thing F. And that's the probability that at a given distance, C, from the sweep site, this is the chance that two randomly chosen chromosomes are identical by descent. That is essentially the same copy. This then gives you an expected pattern as you move away from that sweep target. You can then look at that pattern as you move away from the target and basically ask, is that pattern matching this form or not? And the nice thing about trying to fit this curve, so what you basically do is I go to a certain distance here, that thing gives me a C value from a assumed target. I can then move a different distance, that gives me a different C value. And so what I see is I've got something here that only depends upon the strength of selection, and then something here that changes with distance. The nice thing about these methods is you can estimate the actual strength of selection. And there have been a number of methods that basically use this approach. Wolfgang Stephan is one of the people that push these methods fairly hard. And so Wolfgang and Kim propose what's called the Composite Likelihood Ratio Test. And basically what they do is they compute this likelihood at a series of markers, and then using those series of markers, find out if there's a best fit and where the putative sweep site is and what its strength of selection is. So it's a likelihood ratio approach. The details are given in the book. Jensen found out that this isn't really robust to population structure. And why? Because what Stephan originally started with is you take this and see how it modifies the Waterson distribution. The Waterson distribution is that distribution, that site frequency spectrum for an equilibrium neutral population. If your population was not at equilibrium when the sweep occurred, you're not modifying the equilibrium distribution, you're modifying some other distribution. So they proposed that can give you errors. So what they proposed was a goodness of fit test, which the book talks in detail. So you basically run the composite likelihood ratio test. If that's significant, you then do a goodness of fit test. If that's not significant, then you've got good evidence for that. The book talks about that. That's why it's got this rather elaborate name, composite likelihood ratio test, goodness of fit test. The bottom line, the take-home message is what you're doing is you've got a scan across a region of interest. You're using the structure, the genomic structure of how that variation changes to look for a sweep. It's a way of doing these eyeball tests much more formally and putting them to rigorous statistical framework. And you get things like this. That's how the Waterson distribution is modified. That's the likelihood function you sample from. So a modification of that composite likelihood ratio assumes the Waterson distribution. That is, the population was at equilibrium when the sweep occurred, so the site frequency spectrum was an equilibrium frequency spectrum and shifted. Rasmus Nielsen suggested that let's not do that. Let's look at the site frequency spectrum at markers elsewhere, because that will reflect the non-equilibrium structure of the population. Then we'll ask how that distribution would be perturbed by selection, and that approach is called sweep finder. So it's basically composite likelihood ratio, except instead of using the Waterson, using an empirical site frequency spectrum. Another approach, sounds like a superhero or a new type of car, right? XP-CLRT, cross-population composite likelihood ratio test. The Sweep Finder and Composite likelihood ratio test are, I sample a series of polymorphisms from one population. I use that to test for selection in that one population. The cross-population test was motivated by domestication. I've got teosinte here, I've got maize over there. What I then do is I take a chromosomal region, but instead of looking at polymorphism in the maize population, which these other tests would do, I look at that region in teosinte, that region in maize, and I look at allele frequency differences across those and see how that curve varies. And that curve is also going to vary basically following this form here. So the cross-population test is I sample a reference population I assume that hasn't been selected and a selected population. For example, Han Chinese and Tibetans, with the idea that excessive allele frequencies in Tibetans are due to adaptation to elevation. So this is often used in the search for domestication genes. One big issue these tests have is, again, ascertainment. Because the way these tests are usually applied is people don't scan the entire genome and look for these. That is, they don't take the raw data and simply apply the test directly. Rather, they take the raw data and then they look for abnormal signatures, like excessive to G minus D. Then once they find that, then they go back and run the tests on those regions. That is an ascertainment. You have to adjust for that, otherwise your tests are biased. Because that coalescent process is noisy, and you're going to get regions of the genome which have unusual coalescences by chance. They're just outliers from drift, nothing more. But the model here is not looking on outliers, you're assuming you have a normal coalescent structure. So you have to correct for the fact that when you apply this test, the test isn't applied on random data, it's applied on data that you ascertain to be unusual in the first place. And so you can basically adjust for that. So that's kind of the closing lecture on looking for selection by looking at shifts in allele frequencies over time, shifts in allele frequencies over space, or looking at how that pattern of variation either changes within the polymorphism of one population, that's composite likelihood ratio or sweep finder, or changes across populations. We're now going to talk about site frequency spectrum tests and haplotype tests. Before I do, any questions? Let's do this. Oh yeah, go ahead.
I was wondering with those third generation,
Mm-hmm.
the way you test environmental genes, what if you have multiple environmental factors?
Sure, you can put that in the models. So yeah, that's a good point. Yeah, so basically this term here, remember the key term in the model was basically this X beta term. That X beta term means for given observation I, it's simply this, mu plus beta 1 E1 plus beta 2 E2 plus plus beta N En. You can put multiple different environmental factors, and it could be that E2 here is simply E1 squared. That would give you a quadratic term. So this term here gives you the general linear model, which can be, you can have a whole bunch of stuff in it. The problem is that model's too complicated. You need a lot of data to fit it well. So don't get over your skis, is what they often say. Don't make your model more complicated than it needs to be. The reason you do these models is you're asking, is there an environmental association? You're not asking about the detailed structure of that environmental association yet. You want to detect it first, then you maybe want to do a much more elaborate analysis to actually unpack it. So you can handle multiple environmental factors. You can handle nonlinear factors in that chart. Yeah. So what you can do is, that's a good point. You can basically do a principal components analysis on your environmental variables and then extract out a set of independent components and run those. Right. So usually, again, the problem is your model space is vast. So you want to be careful and clever about exploiting it. If you have a vast model space, you can run so many tests, you're going to get something significant by chance. The problem with running so many tests is, as you run more tests, multiple comparisons, corrections become a more and more and more of a burden. So you want to be kind of smart about how you run these tests. Yeah. Sure. Right. Sure. So the good news is Appendix 4 in the book talks all about meta-analysis and false discoveries. Let me give you the kind of cartoon version for that. And again, you can always get the book in your library. You don't have to run out and buy it, right? So the idea is, if you look at the history of multiple comparisons, a lot of it was sort of set by the mindset of drug discovery. And the mindset of drug discovery is, almost all my attempts are going to suck. And occasionally one will be better. So I'm really worried about letting one that's going to suck get through. So I'm incredibly anal about not making any type 1 errors. Don't make any type 1 errors. Whereas in genomics, it's a bit different. In genomics, we've got a large candidate set. We expect a small but non-significant fraction to not be from the null. So Bonferroni is too stringent. The better way to go is to control what's called the false discovery. A discovery is when we declare a test to be significant. A false discovery is a test we declare to be significant that actually is from the null. There's no effect. So false discovery rate, basically, Story and Tibershani and Benjani came up with this idea of, in that set that I declare to be significant, what fraction of that set are false discoveries? So if your false discovery rate is set at 5%, among your FST values, 5% of those you declare to be significant are going to be false discoveries. That's a much cleaner way to control than to worry about, having no false discoveries whatsoever. Because the reason is you expect a number of tests to not be from the null, therefore you expect a significant number of discoveries, and therefore your initial data analysis is to get an enriched set. And the false discovery rate of 5% says that in this set, it's really enriched, that only 5% of those things are potentially in error. So it's a very different mindset. And so the way the way to probably go is to use Story's false discovery rate. The appendix talks about that, but basically it does a slightly better job in that. The other thing you need to be careful, though, is you can't use false discovery rate on QTL data because they're correlated. GWAS data is better because they're uncorrelated. If I've got markers on a chromosome, by adding or subtracting markers at random, I can artificially inflate or deflate the false discovery rate. And John's got a paper in genetics on that. The reference is listed in the appendix on false discovery rates. Yeah, there's a whole bunch of stuff we can talk about. I've kind of slid that over. But literally, almost all the complications we talk about are in the book. That's why it's 1,500 pages long. There's a lot of sidetracks you can go off on.